I will start talking about the general things about the company, the usual introduction, and then Klementyna will take over from me. Anytime, of course, raise your hand if you want to ask a question. The purpose today is for you to learn something from this. That's it. Any questions so far? Everything is clear? Yeah. Okay. Who knows, what do you know about Clarivate? Question from already. Do you know the company Clarivate? Yeah, what is science? Yes, pretty much. It's not what is science. Alright, so what is science is the biggest, let's say, household name in Clarivate's portfolio solutions. But we have also life science solutions, life cycle innovation, products, solutions that were designed for the pharma industry. So our job is to make it adaptable to academics and governments around the world. That's my job, and it's not an easy job to do. But Clarivate is more about science, life sciences, patents, reports, like many areas. So that's the classic slide of innovation and how to accelerate innovation. I won't bore you too much with the details of it. We help all walks of life. Like I said, this solution that we showed, that we're about to show you, Metacore, was designed for the industry. But I think we can have a collaboration with the Akademi. Okay? So this is the suite of solutions that we cover. Can anyone see okay, by the way? It's too bright. Okay? Can you see? Choose which of the lights, huh? Better? Say again?
Make one slide bigger on the computer.
Sorry.
Can you start the slideshow?
Yes.
Thank you.
I apologize. Is it better? So, as far as introduction goes, it's probably the most important slide. So I'm just going to take all the way to the end. That's it. That is our, let's say, end-to-end solution suite. So we cover pretty much everything. All right. Well, the science is the citation index that we all know, we're famous for. That's what we put in there, so people know what we do. But we also cover drug discovery from benchmarking, purposing, to evaluation, to regulatory, to drug development design, strategy, and also commercial optimization and formulation. And the whole life cycle of innovation in life science is covered by what we do. The great thing is we cover everything, but the bad thing is not everybody knows what we do. It's way too big. Today, for you, for the purpose of today's presentation and symposium, that is going to be the part which is much more in sync with the spirit of the conference. Alright? So for your information, for any question you may have on life science and regulation as a whole, we cover it. And the whole point is to reduce your overload. I remember back in the day when I was a PhD student, we didn't have access to all these tools. I mean, what was science back then? It was quite primitive compared to what we have today, I must say. But I wish we did. So the point is to save time, not to spend too much time on information around help you in your R&D, in your research. We're straight to the point. And that's what we do. We gather information, many sources. We have people working every day, analysts reading the papers, and making it easier to access for people like you for better research. That's pretty simple. And that's what the research intelligence cloud is about. Okay, I was talking about Clarivate. What is science? Hotelis, which is the suite that I showed earlier, the big suite with Metacore inside. But the company is buying more companies. I don't know how they do it, but they're still doing it. They bought DRG. Anyone knows what DRG is? DRG is a company that specializes in research assessment reports per country. epidemiology reports as well, and drug marketability. So companies are looking to do clinical trials or sell their drugs in a specific market, a new market, and we give them information. So Clarivate has purchased the Araç. So they have that as well. That's very cool, okay? Even though some pieces of information there could be useful to academia. And then we have all of, let's say, avenues of working together, consulting. Often what happens is, university or a government agency will ask us a piece of information. They won't be able to get everything. We get what they need from each site, and deliver it to them in the report. All right? And that's pretty much it. I'm going to pass the mic to Klementyna. So Klementyna.
Yes. Can you hear me?
Yes, we can hear you great. Would you like to take charge from here?
Yes, thanks. Thank you very much. I will switch off my camera just to have the better transmission. But of course, if there are any questions, let me know. Okay?
Okay, I'll do that.
Good, great, thank you. Okay, so Ahmed introduced you our suite and introduced what we can do practically in the whole cycle of innovation. And today I would like to show you Metacore, but also other solutions based on an asset, based on today as an example, I took Keşfi, which is an asset in oncology area, and it was initially approved as a monotherapy for multiple myeloma. So when you look at the discovery of it, it was discovered first as a natural compound from bacteria by scientists from EMC, BLS.
Hold on,
Yes.
I don't think you see your screen. Sorry. I shared my screen with you.
Okay.
You wanna take over?
Okay. No. Okay. Now can you see it?
Yes, we can see it now.
Okay. So it's basically the next slide after what you showed. So Keşfi was first. It's a natural compound from bacteria. It was discovered in the early 90s, and they published it under the name of epoxytimine. But they left it without any further work. And then the Academy group from Yale came across it. They were looking for anti-cancer compounds, so they were able to make an analog of the epoxytimine. And this inhibits a target called proteasome, and it happens in the industry.
Hold on,
They
hold
were.
on. I don't think we see you. Sorry. Did you take over the presentation?
Yes, I'm sharing my screen with you.
Yeah, but your screen doesn't show here unless I share my screen with you. Oh, there we go.
But now I'm seeing your screen.
Okay. Again.
Can you press escape?
Even down.
Is there a button that says something?
Okay.
You don't see you still see my screen. That's the problem. On the big screen. I see.
But can you open on the big screen your Teams?
Yeah.
On your screen, open Teams. Where are you?
I have it.
Okay, so let us just show your screen and go to the Keşfi slide.
That's okay. We got you. That's it. Sorry.
Okay, perfect.
Can
Okay.
you go back and repeat that slide? Thank you so much. That's it.
Okay, so we start with Keşfi. As I said, it was found as a natural compound from bacteria and was published in the early 90s as epoxytimine. the research was basically left. So then a group from Yale came, they found the research, and they make an analog from ethoxyzimine, and then this analog inhibits a target called proteasome. And it was a great breakthrough. So it came in a company called Proteolix. And around the same time, FDA approved this first proteasome inhibitor under the name Velocade. And the story, of course, goes further. So the Velocade was very effective, but has also side effects. So, for example, there we can have this so-called unmet need. So the drug was good, but there were some side effects. And then the Proteolix optimized the compound to reduce those side effects and launched Tetrolis. And then they were acquired by Onyx, which managed to get the Tetrolis through the full cycle of FDA approval. And it was launched to the patients. And then they were also acquired by Amgen. So this is like a typical framework specific to the biopharmaceutical industry from innovation. And when you look on this whole cycle, you can see the whole cycle of innovation, from research to discovery, preclinical, clinical tests, approvals, and of course commercialization. So today we will concentrate on research and discovery, but we will go also a little bit in the direction of preclinical, and clinical information. And as you know, as a main part today, we will talk about Metacore, which is a quarterly solution that allows you to quickly and accurately analyze multi-omic data, then compared with 100% manually created vessels pulled by a team of scientists. And each molecular collection in a Metacore is annotated with mechanisms, direction, and effect, so that you can quickly comprehend molecular context of disease. And this ultimately allows you to have greater confidence in your decisions, since you can rapidly generate and validate your hypothesis. And we call Metacore your GPS in pathway analysis. It gives you a molecular understanding of disease. You can analyze and understand experimental findings, so your omics data and data from others in the context this pattern of validated biological pathway. So you can, with help of Metacore, you can generate and confirm your hypothesis for novel biomarkers, target mechanisms of action. And the content from Metacore comes from peer-reviewed articles, and all of our data is manually curated by a team of scientists. So they are PhD and MD level research professionals with, I think, more than 25 years of experience in the industry. They have a few scientific expertise from medical chemistry, molecular biology, and more. So each editor undergoes strict training, and so finally, that gives you the possibility to give you the high quality of data. So the data, as I said, published from peer-reviewed journals like Nature or PLS one. And each molecular interaction is annotated with mechanism, direction, and effect, so that you can quickly comprehend the molecular content of the disease. And this, we have now more than 0.6 billion molecular interactions, over 1,500 molecular pathways, and over 270,000 gene variants. So you can use Metacore to validate your data, to your hypothesis, but also Metacore is a great tool for knowledge mining. And so with Metacore, you can compare, contrast, and visualize workflows for data sets. And as an example, because the idea is to show you how Metacore works. So the idea today shows you the text how to work with Metacore, and API, you can go on the website, and here you have Ahmed ID number and dataset number, so you can download the data. It's an up data, because Metacore also allows you to analyze data your own, so you can upload the data to your experiment and also from others. So here we have, in this data set, we have gene expression data from multiple myeloma cell lines, and the hypothesis is associated with multiple myeloma. And here the tells two different lines were exposed to stepwise increasing concentrations of the medicament, particles of it, over a period of 18 weeks. And the cells adapted to growth by 4 weeks and different concentrations in different times. Yeah? So the resulting cells, they retain resistance to the drug when they were tested after removal of selective pressure from the time was, I think, about 8 weeks of the experiment. So the main conclusion from the data, so here we see the data, the p-value, the test threshold, was that protease inhibition induced the accumulation of that protein in the intracellular environment, causing over PR overall and stress. So it's basically about the cell apoptosis. And when we have this data, now we will go to live demo. So I prepared it already for you. And now on your screen, maybe Ahmed, you can confirm, you can see the Metacore surface. Can you see the Metacore? Yes? Okay, perfect.
We can. Yeah.
Okay, so great. So Metacore, you work under your private login and password. So all data that you upload, every analysis that you do in Metacore are strictly private, so nobody has access to those. And then when you enter Metacore, you can see here we have like 4 different areas here. Here you can see your data. I already have some data here, so you can upload your own data using tools here, and you can also share data with others. So even if you work privately in your Metacore, then you can also share data with others. And I have this data that I'm talking about today from the petrolysis experiment. You have here these 2 data sets. And you can, you have a list of activated data sets. You can activate up to 4 different data sets. So you can also compare the data sets, the different experiments up to 4. And each data that is yellow, green, dot, it means that this data set is activated. And here on the left, on the right side, you have different activities, different possibilities that Metacore gives you. So you can use it as a knowledge mining tool. So here you have a section of content, and below, every time when you choose one area, you get explanations. What does it mean? What you can do this, and how this tool works. Here you can have different workflows. We have one-click analyzers. We have also here a possibility to upload our data, and as you can see, you can upload different kind of data and have analyzed here. So when you start, as I said, you can start as a mining tool. So here, for example, you can search for you can use easy search or advanced search. You can search by ontology, for example, for pathway maps. And here you can start with myeloma, multiple myeloma. So you can start with easy search just to see what data we have. What data we have here, and as you can see on the right side, we have an object fund, so you can see what genes are associated or found in the data, and extracted from different peer-reviewed journals. So the specialists, they are looking on the pathways, they are looking on the information in the publications, and then they upload the data as upload here. uploaded here. So you have different proteins and so on. So you can start from here. You can also the components and add it to your analysis. So this is like a knowledge mining, very powerful. What I wanted to show you how we work with your experimental data. So we have already data in our in the system. I activated two different data sets, and the first question that I want to present to that Metacore helps you to answer the first point is how to identification of the signaling pathways that are statistically enriched and differentially expressed. So differentially expressed genes in this drug-resistant multiple myeloma cell lung. So for this, when we go here on the workflow and records, here we will have a look on enrichment analysis. So you can, as I said, you can activate up to 4 different data sets. We have 2, and then when you click on the enrichment analysis, you can pull here more data, or if you haven't analyzed any data, you can do it here as well. Then you click on next. I already prepared some analysis here so that you can then see it later, but that we don't have to wait. But as you can see here, we have our data sets, and we have here our data. There's our human cell lines that we have here in Metacore. You can also have data from mice and rats. And then when you can upload, you can set threshold for your data set if you need it. And then when you click on apply, Metacore will calculate the pathways, and will look on process network diseases. So everything what could be found with the gene from the cell lines from our data. And you can see how many network objects you have. So you can these data sets are not so big, but you can have really huge data sets with thousands of objects workflow. I chose this one because then we get the results quite fast. We don't have to wait, but Metacore works very fast. So even with a bigger data set, it's not long. So now when you look at the data, it's calculating one by one the points we have diseases. Now the gene ontologist processes are calculated. And when we look here on the upper part, you can see. Okay, now it's done. Everything is calculated, so as you can see, it was quite smooth. And here, on the first slide, we have the pathway maps. For our 2 data sets, they are marked in colors, so yellow and blue. And here on the graphs, you can see how many genes from the active data sets are in the pathway. So here you have the numbers in the graph, but also here on the right side, you can see for the genes in the pathway how many are in the data set. So from the kinase 11, we have here 8 genes, and for 31, and for the kinase 34 cell lines, we have 5 genes. So in this way, you can compare it, you can see it, and those are different maps, so pathway maps. So the data that I uploaded is compared with the whole data that we have in the system, and that you can where the genes were also find with the genes that we have in our data sets, in which pathways of these were found. And you can here you can extend the number up to 500. So I put now on 100 because somewhere there we have also pathway map for multiple myeloma. So in a moment we will see the longer list. And the same is like for the process networks, for the diseases. So here you can have a look on different diseases. And now here when we go down, here you can see further points. And here on the number 18, actually we have a pathway map associated with the multiple myeloma. So you can click on the name of the pathway map, and then you will be taken to this pathway map. And first you saw the whole pathway map that we have for this disease. And now you can see actually like not grayed up, but sharp in color those genes that are from our data set. They are, you can see which are active. As I said, one data set it was 8, from the other was the multiple myeloma. I haven't looked on the number. So here we had a 4 and 6, right? So here you can see those. And this pathway map shows you also for each of the genes if it's down-regulated or if it's up-regulated. So here you can see for this, we have 2 data sets active, so you have for both of them, if there was regulation down or up. You can see the like thermometers. So everything what is blue, what goes down is down regulated. This thing is down regulated. And here we have also gene which is up-regulated from the second data set. And when you hover over the object in the map, you can see the information. You can see also the value here. And the whole map shows you also, like when you look on the here, we have the cell. We have here the map starts from the top. And what is great here in Metacore, you get not only the information here in the form of map, but you can click on each object in the map, and in an extra website, you will get additional information about this. Here we have citing D1. So that the symbols and the colors, they are characteristic for all. They are common for all maps, so you can easily see which information we have here, which object. And when you want to click here on the legend, also in the extra window, you will open the legend, and you can see that here we have a generic binding protein, because cyclin D1 is a generic binding protein. Here you have information, so you have chromosome location. You have description, you have also the ontologies from, as I said, here in Metacore we have data from human, rat, and mouse. And here also you can see the gene bank information, publication information. So you have, when you click on the object, you get the whole information behind, whole knowledge behind that it's under cover of this map and whole and all of it. And also you can see the direction of the interaction between the object. You can see the direction, but also what kind of interaction here. So here we have regulation. Here we have influence of languages. You can see the direction, the effect, and also mechanism of each action. So this gives you information practically like from each pathway you can see this information. And also when you go here on the disease, you will get also mechanism. You can also sort search here for the information. So when you click on somewhere here we have lung plasma. So you click on the disease, you will get information on the disease. And also here from this topic, you can have a look on pathway maps that are associated with the condition. So you can go like step by step, discover more and more information, which normally you will really need a lot of time to go through the whole literature to find this information. Okay, so this was the enrichment analysis. So this is where we had the, as I said, where we had the maps. You can also click on that part of the class, and you can see this information there actually additionally, and also you can click. So when you come back here to the pathway map, for example, you can click on the numbers here, and you can see the network objects. So which objects are there, so the tables that are active in this particular pathway map from our data set, and you can use them to build your own network, and you can export them as well. So you can like find that information, and then you use this information for further analysis. So this was the enrichment analysis. So here we could see how you can identify signality pathways that are statistically enriched with differentially expressed genes in our cell plan. Okay, so this was one of the analysis. And the second analysis that I would like to show you is the possibility the Metacore gives you comparing your experiment. So I would like now to explore common and unique mechanisms of drug resistance with those two cell lines, just to get insights into different mechanisms of fibrosis resistance from those two cell lines. So for this, we are here also in the workflow, and I'm choosing here the possibility to compare experiments. When I go through which analysis, which workflow I would like to choose, you can always click on the question mark, and it will open you extra page with help, where you can step by step see what does it mean, what can you analyze with this kind of analysis, what data you need for that. So here you get every single point is described. So here you have very extended help. And here, of course, below you have short explanation, but if you need more, you can just click on the question mark. Okay, so now we would like to compare those two experiments, and when we I have it here, but it's going quite fast. Yeah. So again, we have here our cell lines, and now we are comparing experiments. I click on apply, and system Metacore is now comparing those two data sets. So it's looking basically for which unique the mechanism we have there, which gets unique there, and you get the result. You have a graph, the comparison result, and then Metacore calculate the pathway map, code processes, process network, and all this information, but now as a comparison. So here, the first graph that you get on the comparison results, this graph shows me unique. Here those are unique, so jumping because it's calculating. So here you have unique, similar, and common genes in the compared data set. So also here I will wait till it's calculated. Also here you can click on the bar so that you can export here, for example, the unique genes. Here we have so here we have the total network object. Here we have the information. So when we click on the number, also in extra point we can export it from this is niches, and we can use them to build our own network. We will do it in a moment, but in this way you can see this information. And so here you can also click on the common and also go. So each part, each component you can export and use for further analysis. And here below we have, as I said, we have a pathway maps, genome ontologies, and here you have also, when we look on the somewhere here we have our multiple myeloma path, and what is here very good in Metacore, also in the other the results that we get. But you can also filter by different categories. So by different, in case of pathway maps, you can look on different map categories. So here we have disease maps, for example, and when we go here to neoplasma, somewhere here we have multiple myeloma. So you can deselect also here just to get if you are looking on a specific condition, so that you don't put on everything and you don't get distracted with too much information. You can narrow down to this condition that you are interested in, condition or metabolic map or regulatory map, depending what you need. And when you click apply, you will it's still generating network, so it's still working in the background. But in this way, you can easily concentrate on experimental data from this that you are interested in. So it's calculating. It's working because I went there, so I added extra filters, so it's still calculating. But I think that in this way you get already idea how it works and what you can get there. And of course also here you can click on the number. You can see the information on the p-value. So I think I will skip because I think it looks a bit longer. But also here, as I told you, like we have also a map about apostrophes. So in this way, also here you can see the map, and you can actually see where exactly how this pathway is working. And as I said, the whole details are similar in all maps, so that you can easily follow it. And this map you can use, for example, for your presentation, for your publication. So this is a visualization as to visualize your data. Okay, so the last part, the last experiment that I would like to show you is building your own map. And this build network. So here you can also go to one-click analysis, and you will have single pathway maps and everything. But now I would like to show you how to build your own map from the data. As I said, you can upload the data. You can have your own experimental data. So here you need to choose a network for your experimental data. For these purposes, I will activate only one data set. And here you click on Build Network for Experimental Data. Here again you can choose the data that you want to use, and then you can have a look here on it's working. I already opened it here to have it a bit faster for you. But then you will come here from this point, you will come to the network options. So there are different algorithms that you can use for to build your pathway map. Depending on here you have always explanation. So depending on the number of genes in your data, and also of course on the purpose. So what is your hypothesis? So what would you like to find out? So what would you like to visualize? You can use different different algorithms. I would like to show you which differentiated genes are regulated by the transcription factor KALF4, which was suggested that it plays an important role in the drug resistance. So for this purpose, we will choose the shortest pathway, and here you can decide how many steps your pathway will have. So I would like to see that the direct regulation of the transcription factor KALF4. That's why I'm putting the number of steps in my path too much. So I want to see only the direct regulation. And then you can use also to build your map. You can include in your map, in your network, canonical pathways. It takes them longer, so be aware. It's good to try one with canonical and one without, depending on your experimental data and what hypothesis you would like to validate. But now I choose not to use the canonical pathway. And you have here also additional options here. I already selected some. One of the type of the interaction you can set here. And as I said, I'm looking at transcription regulation. That's why I choose only this single interaction. You can also choose here tissues, cell lines. We are working with the human cell lines. You can choose a species. As I said, we have here from human, mouse, and from rat. You can choose your orthologies, and you can choose object type. Here you have, so this is the additional option. You can decide here if you want to see disconnected synod or not. And also you have the possibility to have a look and decide which objects are in your network. And I would like to start with the KALF4 transcription factor. So my network should start, so my interaction should start from this object, and not going through, but they are supposed to go to and from. So what is the regulation? What is which are regulated by this? That's why I have starting from this point and to all other objects that I put here. And then you click on build a network. I took the advantage of being prepared, and I already prepared the network for you. So after when you calculate the network, you will get a huge network, and everything starts by Ideal 4. So here you can choose sorry, it's not responding so fast, but you can. Okay, it's loading again. I hope it will load everything. So you can have a look on the. Click on the. Okay, so here you can zoom in just to see particular parts of the network that you have here. So in the center we have KALF4 factor. And here, as you can see, we have this quite busy network, many objects, but the thing is that the more research is done on disease, on a specific drug, for example, and we put more and more information in the Metacore system. So the data is the upload of the data is on a regular basis. I did this network less than a year ago, and it had way less objects than it is now, because there was a new research done on this project, this condition, and also on this drug. That's why I have now like the network is getting busier and busier. But and this is very easy visualization of the processes that we had before on the pathway maps. You can see the interaction. So everything what is red. Red points that this is it's inhibition. The green arrows shows us, for example, here the green arrow shows us activation, and also every network object with the red circle around. This is the end point of the interaction, and I thought only once that interaction. Why we have our color factor in the middle, and the red are all of the objects are with red circles because they are the end points. And as I said, the green arrow is the activation, red is inhibition, but we have also unspecified action. So we have here gray arrows. So there are many gray arrows because we don't know yet if it inhibits or if it's activation. When you look on the objects, you can move those objects. So you can put the network if you are interested specifically in one object. You can move this object on the side so that you have better view of this object. And as you can see also, for each object we have here extra information, so we can see here if it's upregulated or if it's downregulated. And also here you can click on each network object just to get information. As you can see, my browser or my connection is a bit delayed. That's why I'm moving moving the objects, and they are moving after a moment. This is so this is how your object, how your network can look. But of course, you can work with the network layout. You can change. You can move the objects. You can also decide which interaction, which information you will see on the network. From default, because I choose the transcription regulation, there is on the arrows, we do not have information what kind of mechanism is this, or interaction mechanism, but you can click and you can choose to see it, or if you don't want to see on your future network, you can deselect so that you cannot see. After a moment, we will see that now every arrow has a small sign, and this is I will try to zoom in, but there is a TR, so transcription regulation. And when you hover with the mouse, also here, when you hover with the mouse over an interaction or an object, in the left-hand corner, you will see the information. What is the information between which objects, what is the mechanism, what is the effect, and what is the trust, so if it's corrected data or not. So here you can see all those information. And you can also here and go and see the different information. And also here you can change how the network is shown, so that you can have like more close to the center, because you have a one central point, but you can change the network layout. It takes a moment though to rebuild in different topology. And you can, as you can see now you can see exactly where the process that we are talking about here, that we have here in the network, can be placed. Where is the extracellular, where is the membrane, cytoplasm, nucleus, and which are unspecified. So we know exactly where all those processes are taking place. And you can also filter interactions. So you can decide which one you will show. If you want to see the unspecified report, so you can hide them or show. And also you can when we are here, and you can filter for network objects, and you can decide which one you would like to see from which experiment, which basis. And when you are go here on the genealogic processes, I told you that this the glycolysis was the whole process was about also the apoptosis of the cell connected with the apoptosis of the cell. So when we look at the regulation of apoptotic processes, you can select those and can show those interactions in your network. So you have a very like a wide, I think, actually endless possibilities to set your network and to value your hypothesis. In a moment, it will when you click on trace. In a moment, they will those that are connected with regulation of apoptotic processes. They will see. They are now darkened so that you exactly can see which objects, so which objects are regulated by the transcription factor Klementyna in the apoptotic process. And you can of course switch now. You can see it in the centric point, and you can, as I said, this is the network. So you can build endless networks in Metacore. You can export those. You can use them for your page is not export. Sorry. So you can see the information here, and you can there is some with my connection. So you can see can use the network for your publication, for your presentation, and you can also export it, and you can have a look. Like you can save the data, so you can work on it later. Also, as you can see, like when you start once you have a hypothesis with one data, after a time when the new data is uploaded in Metacore, you will see more information on your processes, on your network. So it's when you work constantly on a project, it's good to come back and recalculate the network to see if there is a new data uploaded, if you can get some new insights. Okay, so this is what I wanted to show you for the Metacore. Just to summarize, Metacore helps you through looking on the molecular basis of disease. It helps you to visualize how your target works and how your drug could have impact when you are working, for example, on a drug. But it may be also helpful. So we are here on the research and discovery, but also may be helpful in the further in the clinical part when you are looking for example, on this on side effects. So what is the mechanism behind the side effect? And with Metacore, you can examine if there is evidence of genetic link between the target that you are choosing for your experiment and the given disease. You gain a molecular understanding of the disease mechanism as well as the pathways and processes impacted by your candidate, drug that you are working on. And you can really accelerate your discovery of disease, of the toxicity of biomarkers, and analyze their potential. And you can really understand the scientific knowledge. So you get the understanding and the whole scientific knowledge around the biological process and molecules of your interest. So you don't have to look in many resources, but you have everything together. So I get often asked what makes Metacore different? And there are, just to summarize, several ways. So the 100%, remember that the 100% of the data is reviewed and input by a team of specialists to understand the nuances of early research. There are no restriction on the type of data, integration of data, or size of the network you can build with Metacore. So you can build really busy networks. You can share your data with other Metacore users to easy communicate. And unlike other databases, you will find mechanisms and what is important, the direction of the interaction, and also effects linked to each and every molecular interaction, so that you don't have to go, as I said, to other resources and look for the information. And finally, Metacore is fully integrated with the critical data you need, including gene variant information, just to make your that will help you make well-informed and data-driven decisions. So that's all what I wanted to show you from the Metacore. And we were talking also about the course. So Ahmed already introduced you to Metacore solution. And this is a whole suite of different solutions that we have, the source of comprehensive, validated, selected, regularly updated data and analysis on researcher biological pathways, protocols, facilities, patient populations, global regulatory requirements. So it delivers really insights from different points of information, hidden data, and helps you design your research. And there are different components. And one of the components that is really helpful in the type of the research and discovery is drug research advisor target drug ability. It was designed to assist researchers to quickly identify and run potential targets for disease or condition, because you need to know when you are working on molecule, when you are working on a new drug, you need to have a target for this drug, and you need to be sure that this target really works. And you can, and with Metacore, you can investigate the target uniqueness, its competitiveness, and to get the confidence whether your target is one that you should move forward with. If there are others working with this target, or how many other drugs are there in the active development, so that before your institution makes big investment in your research, so that you can really be sure that this research will not have, for example, too much competence. So you can search for condition by drug or by drug to see, as I said, what other drugs are there in the pipeline, and then the target drug ability provides you biological evidence. For example, information also on the specific pathway. So it's also some extension and addition for your research to Metacore. So when we go now live to okay, look out. So I need to give my password there. It's also, I think what you put there is it's secured with your password. So you need to have access. And when you start with the drug research advisor, you can start with the condition. So let's say myeloma. So when we search, for example, for multiple myeloma, we click on export, and already you can see that we have the whole condition for the whole disease. We have more than 1,300 targets. So it's very high list of the targets, but you can choose different filters. So you can decide what kind of target you would like to have, with protein or RNA, what are the targets. You can look on different target family, different pathway maps, so you can have information that you are looking on different specific pathway. You can have editor, drugs, and so on. So you have many possibilities, and you can click also on the names here and see information about the specific target. but let's go a little bit more specific. So you could search the drug, but I would like to show you the PSM B5. This is as I told you, hydrolysis is proteasome inhibitor, and the proteasome itself is quite complex. So we will look on a specific subunit which is targeted by hydrolysis, so one of the subunits. And when we explore there, then you get we have two targets. We have a target here, as you can see. And it's very interesting because I was looking for that earlier, and there was only one. So we had an update, and now we have another. Everything what is in green, we have two different targets, and you can see one of those in green. There is already launched drug this there. And here we have also phase 2, so there is some new target for phase 2. When you click on the enzymes, you can see those here, and you can choose one of them, or if there are many, you can choose one by one. And here we have we call it the Sanborn visualization, which shows you the relationship between conditions and the evidence. So when you hover over, you can see the other indication. So what we have here, it's not only multiple myeloma, but there are also different other conditions. And then here you can click on one of those parts and see information exactly how many drugs you have, how many biomarkers. But what is important here is the drug research advisor gives you the biological evidence. So if you evaluate target and you are about to do investment, as I said, you want to be sure that you have a good target. So here you can click on genetic evidence citation. So here you will open you can have access here to the literature behind, so that the genetic evidence is very important for your research. And you can also have a look here on the table. So not only as a sample, also here we have a list of the conditions, and here we have multiple myeloma. You can of course set also here. And here you can see for multiple myeloma, we have 6 drugs in the pipeline. And when you click on the number, then you will be taken here to the information of those drugs. And here you can click on it, is the name of the cartridge, or the cartridge. You can see that it's launched, and you can see that there are already some other that are biological tested or in the preclinical, so you can see the page. So we know now that if I was doing the research on this, I would be sure, okay, I will not be the first one, but the first in class, but maybe I will not be the first, but I could be maybe better. So that gives me additional information and helps you just to understand information. And of course, also here you have the pathway map. So you can see the pathway map, and here, for example, you can see anti-apoptotic pathways, multiple myeloma. So you can click on the map, and as you can see, it's quite like the maps from Metacore. So you have also here the map here, and you can see information, and also here you have interaction effects and so on. But then you can move now back to Metacore and have a look more specifically on this map if you are with this map interested. Okay, Ahmed, question to you. Do I proceed further with the Cortelis?
Further than Cortelis, well, first of all, question for the audience. How are you? Are you okay? Can you hold on? Or are you? By the way, I was told it's not very clear on the screen. It's like the other time. No. Okay. Sorry? Resolution? It's not this, I think it's the connection. What time is it?
unfortunately.
Okay. Okay, does anyone have any questions or any comments? Have you learned anything? Or is it all blur? So?
Was it interesting?
It was interesting, but you see, the issue was the screen wasn't clear, especially with
Okay.
yeah, exciting. So I think we should maybe call it for the end of it, Klementyna.
Okay.
I'll gather further feedback now, yeah, we'll take it from there next time. Okay?
Okay, great. Thank you very
Let me
much.
show, you can show, yeah, perhaps you can show yourself and say goodbye before we see you. There you go.
Yeah. Hello. Yeah. So thank you very much for your time and for interest. If you have any more questions, or we will provide you with the presentation so that you can get some additional details, and also on the portfolio. And if you are interested in presentation, maybe more clear screen of the Metacore or portfolio or target drug ability, let us know. We will be happy to show it to you. Thank you.
Thank you, Klementyna. Thanks so much for your help. Take care.
Okay, thank you. Have a great afternoon. Thanks, bye.
Alright. Okay, I think we can. Okay, I want to thank all of you for staying that to the end. I sincerely apologize for the lack of technical savviness on my side. It was not easy fitting in a non-computer design drug development product into such a conference, but I hope you learned something. We will follow up with you in the near future. For any questions, reach out to us, to my colleagues from online meeting, and hopefully we can